I think I'll start again. People can filter in as I talk. So you already saw that I showed this phenomena of persistent spiking to you. It was discovered by Angel Alonso. Sadly, he died in 2005, but so he hasn't seen the further development of the field. But he did really exciting work on this topic. And if you think about it, this is really interesting in terms of a potential mechanism for working memory. All right, so it's very standard. We talked a little bit about working memory being a potential mediated by sustained activity, and in the delayed match to sample task, you could argue about whether or not it's a working memory task, but you can think of it as involving the holding online of a sensory stimulus. In this case, the monkey sees the color of this dot, and then he sees two dots. In this case, it's a delayed match to sample. He sees a red one and a green one, and he has to respond to the red dot. So he's holding this information online with the intention of making a response. And as Fuster showed, and many others, like Earl Miller and so on, Niki, various researchers have shown that there's often a response to the sample stimulus that can be maintained during the delay period and seems to be relevant to the matching function, and then often will turn, come back down again after the match. All right? And as I mentioned before, the standard kind of view of this sustained activity is that it involves attractor dynamics with excitatory feedback. So there's many models out there that have used neurons with excitatory feedback connections, usually in prefrontal cortex, and these excitatory feedback connections maintain an attractor state even when the input is no longer there. So the input activates the circuit, and then the activity during the delay period is this reverberating excitatory activity. But this persistent spiking is different. I said that happens even when synaptic transmission is blocked, right? So instead, it's an internal process, this interplay of the CAN current, this calcium-activated cation current, and the calcium influx that maintains the spiking even if you block all the excitatory transmission. But it's a different way to hold on to that activity. And it would be particularly useful for novel stimuli, because with novel stimuli, you might not have a previous existing set of connections to mediate the working memory, but you can still hold it online. And this means that novel stimuli might be particularly sensitive to block with acetylcholine receptor blockers like scopolamine. And I wanted to relate this to some work that Trevor Robbins did, actually showing that scopolamine actually impairs human working memory. And there's a few other studies showing these sorts of impairments. So it could be contributing to that impairment.
Sorry, this is a little bit outside of, so it's probably a very stupid question, but I understand the idea of the persistent spiking, but what actually stops that? So when the agonist is applied, for example, what's the mechanism that
Yeah, that turns it off, you mean? Yeah. No, that's a really good question, and it's not completely clear. So there was at least one paper from Angel Alonso's lab showing that when a match-type stimulus, a second depolarizing stimulus coming in, could turn off the persistent spiking in certain neurons. All right, but then there's other papers where consecutive depolarizing inputs would gradually push up the frequency of the persistent spiking. So there's a lot of details about it that aren't clear. But the idea with the depolarization is that the second depolarization can cause enough activation of the calcium-dependent potassium current that the balance shifts towards the down state, in a sense.
Can I just follow this? Sorry. Can I just follow this up, Mike? If you translate this into a behavioral situation, can you envision that persistent spiking goes on, let's say, for 15, 20 seconds to mediate intertrial intervals? Is that reasonable?
Yeah, so I think that is reasonable. Certainly in the slice, it can last for many minutes, so it's a pretty robust enhancement. And so I think it could be the mechanism for these 15-second delay periods, at least.
There was a paper published a lifetime ago with Bertinoro looking at in vitro in the frontal cortex. And there was an interneuron that would latch, where you give it a depolarizing pulse, and it would keep firing until you gave it a hyperpolarizing pulse, and then it reset the baseline state.
Yeah.
So it seemed like the polarization started and the hyperpolarization ended.
Yeah, so that's another way. I think I had the slide in here, but I took it out. Let me just see if I don't think I have that one in here. No. So, but yeah, so that what you say, so there's either mechanism can be used, either a second depolarization or a second hyperpolarization. So it probably depends on kind of the parameters of the interaction. But just to kind of reiterate what I just said, so the traditional model of familiar working memory, like in the two-back task, is that the idea that excitatory recurrent connections mediate that sustained activity in prefrontal and parietal cortex. But what I'm proposing is that this intrinsic mechanism could contribute to the maintenance of working memory, for instance, in temporal regions, but it may also work and contribute in prefrontal regions. And there is some evidence for working memory for novel stimuli in temporal lobe structures, which we review in this paper in Trends in Cognitive Science.
Yes, because, frontal working memory is very subject to interference.
Yeah, that's an interesting...
Going back to your earlier point.
Yeah, if you lesion the prefrontal cortex, you get severe, problems with remembering highly interfering small sets of stimuli. So, and that we review that as well, this work from Gaffin's lab and so on.
So what would happen if once you did this, you stimulate with some input and then it keeps firing? If you had another input to that, what happens?
Yeah, so in the slice you can actually get away with that, because the cells are kind of functioning independent of each other, but you're kind of thinking about like working memory capacity, things of that sort. And there are models that have dealt with that, like John Listman has a model that essentially uses a mechanism like this, an after depolarization, to maintain neurons on different phases of the theta rhythm cycle. And he argues that the capacity of the working memory is dependent on the number of gamma cycles that you can fit within a theta cycle. So that's one potential way in which you could store multiple different working memory, or multiple different items in working memory. So I'm going to just skim through this because I'm going to run out of time, I know, and I definitely want to get to the consolidation work. But I just wanted to say that we work with Erik Franzén, who's in Stockholm, has shown that essentially this persistent spiking can be modeled based on the interaction between the CAN current, this cation current that's dependent. This is showing the dependence of that current on the calcium concentration, all right, the intracellular calcium concentration, and a balance of that with the calcium-dependent potassium current, which has a different, in the model, we're assuming a different function dependent on calcium. And so you can see that when the calcium levels are low here, right, the CAN current is more active and it pushes the membrane potential up, but then when it reaches a certain point, the activation of potassium current balances it, and that's why you can get stable persistent spiking. If you only have the CAN current, if you don't have this potassium current, then you'll actually get acceleration of spiking. And we've just used this model to model the delay period activity that was shown in entorhinal cortex by work by people such as Howard Eichenbaum as well as Wendy Suzuki. But I wanted to connect it to Martin's work. So in this review paper, we actually proposed that the persistent spiking could contribute to the influence of the acetylcholine pulse on the subsequent response, with the idea that there'd be, thalamic input coming in, and these feedback mechanisms could contribute to the creation of this cholinergic pulse, and that could facilitate the response potentially by causing persistent firing, rapidly inducing persistent firing due to this conjunction now of the glutamatergic and the cholinergic input. All right, so that's, you can go back and look at this review paper and see the ideas behind that. All right, so, but I mentioned how it could be selectively important for novel stimuli, and I talked about some of the data in humans and basically on working memory. We've actually looked at this idea of it potentially being more important for novel stimuli. We did a delayed non-matched to sample task in rats with cholinergic lesions of the innervation of the entorhinal cortex. This was work done by Jill McGahey, who actually also worked with Martin for her graduate work. And she showed that there was an impairment of delayed non-matched to sample performance for novel odors in these animals that then would go away as these odors became familiar. So that supports the idea of this importance for working memory for novel stimuli. And there's also work I've done in collaboration with my wife, Chantal Stern, as well as Karen Schoen at Boston University, showing delay period activity in a delayed match to sample task in human subjects looking at fMRI, particularly during the delay period, and seeing activity in parahippocampal regions and then correlating that delay period activity with the subsequent memory for the stimuli tested at the end of the experiment, so testing the encoding of that. And this activity associated with that subsequent encoding during the delay period was particularly present in perirhinal cortex and entorhinal cortex, suggesting that there's some maintenance of these stimuli that could be important for encoding the stimuli.
Since the acetylcholine pulse were longer, could you have a larger delay and still have the memory so that the working memory span will increase?
Yeah, that's a good question. So you're saying if the pulse of acetylcholine is longer, you're going to store more different items or more information. Is that what you're saying? Or?
That or even if the sample was presented a longer time ago, the memory is still persisting so that even if the match comes much later, it's still there.
Yeah, I mean, it might provide a way to make the sample maintenance more robust as well. So it'd be interesting to see. And you do need to have these samples present for enough time to presumably activate whatever mechanism is being involved in maintaining them. But then we showed in this study that this activity is reduced by scopolamine. So we actually see a reduction in this delay period activity in the subjects that have been given scopolamine. So it's suggestive of the idea that acetylcholine muscarinic receptor activation is contributing to this delay period activity in human subjects. There's obviously, it's indirect in the sense of this systemic injection, but it's supporting this basic idea. All right, so that was persistent spiking. I want to briefly mention, I'm going to go relatively quickly through this, but I want to mention that a very important target of neuromodulatory substances within cortical circuits are the inhibitory interneurons. And I haven't really talked about them less. You can see I sort of extended my list to add modulation of inhibition here, but I think it's a tremendously important effect. In fact, all of the modulators cause quite robust depolarization of particular subtypes of interneurons in cortical structures. So they'll cause a really robust depolarization in many types, but they'll also surprisingly cause presynaptic inhibition of GABAergic transmission. And this is, again, something that's been shown in a lot of different studies. This is just showing one case. This was work done by Babak Tavaldari and David McCormick's lab in a collaboration. But essentially, it's showing the different cell types in entorhinal cortex, stellate cells, pyramidal cells, but then also labeled subtypes that Babak Tavaldari labeled for somatostatin, parvalbumin, 5-HT3A, vasoactive intestinal protein, neuropeptide Y, these different subtypes that can be labeled with different markers. And these different subtypes seem to have different sensitivity to different modulators. Obviously, this subtype are the cells that have the ionotropic 5-HT3 receptor, so they show robust depolarization with serotonin, very important modulatory effect. But there's also selective effects with cholinergic modulation, particularly with somatostatin and VIP neurons. And it's also modulating transmission. So this is just quickly showing inhibitory synaptic currents, actually, that are being recorded in whole cell patch, showing that we get these synaptic currents, a fast GABA A current and a slower GABA B current. And when we put carbachol into the slice chamber, we see suppression, so essentially presynaptic inhibition of these GABAergic potentials. And this is something that's been shown now in a number of structures, Pittler and Alger, Behrens and ten Bruggenkate, a number of people have shown simultaneously depolarization of interneurons by cholinergic muscarinic receptors. So you have increased interneuron spiking coupled with suppression of evoked inhibitory synaptic currents. So more depolarized neurons, but less GABAergic transmission, which is puzzling initially. I don't have the final word on that, but I put this into these simple attractor dynamic models to look at the effect on the attractor states. And I got what Trevor showed earlier, which was this curve where you could imagine that it's essentially kind of enhancing signal to noise ratio. So you have a baseline response to different levels of depolarization to the glutamatergic neurons. And when you change, you depolarize the interneurons, but you decrease their transmission. Because you're depolarizing them, they're actually causing more inhibition in the default state. So you actually get inhibition of the background activity, but because the feedback transmission is getting weaker, when you get a superthreshold state, you can actually get a stronger response. So this is a way of thinking about it as enhancing the signal-to-noise ratio, which is relatively simple framework, but it's a framework that's out there. You might hear about it again in terms of norepinephrine as well. Just briefly, I want to mention with this cholinergic modulation, there's exciting recent work now from Michael Stryker's lab. Chris Neal discovered this effect initially, and then Fu followed up on the mechanisms, where what they were doing is they had mice running at different speeds. So this is just the running speed of the mouse, a head-fixed mouse running on running like a sphere. And the speeds going up and down. And they showed that, initially it had been shown that the response of visual cortical neurons would change dependent on the running speed of the animal. So there was some change in the circuit that made the neurons respond. The visual responses are stronger when the animal is running versus when it's stationary. And what this study showed was that the VIP neurons in visual cortex are more active, all right? This is imaging work, but they're more active in correlation with the running period. You can see the correlation here, as opposed to the non-VIP neurons not showing that tight correlation. So they're showing these VIP neurons showing quite tight correlation, maybe a bit with PV, less with SST, and then they could block this correlation by giving, and this is, so unfortunately it's the same three colors, but now the three colors are showing three different conditions where they're essentially blocking nicotinic receptors to show that this activation of the VIP neurons seems to be a nicotinic input. All right, so that's quite exciting that this mechanism for the running effect could be due to nicotinic cholinergic modulation. And there's a number of groups that are showing similar effects. Adam Kepich's lab showed something similar where they showed that a stimulus actually in a particular task would activate VIP neurons at a particular rapid interval, and then they would see inhibition of other neurons, which suggests maybe they propose that the VIP neurons are inhibiting the somatostatin and the parvalbumin neurons. And then they'd see a delay activation, which they believe is a disinhibition of the pyramidal cells. And this, in the context of the Stryker data, could explain why the pyramidal cells would then respond more strongly to visual inputs. So this is an exciting new direction of research where a lot of researchers have come into this field to work on this because the running of the mouse is a relatively easy behavior to manipulate the state, essentially, of the animal. In a sense, maybe going from active waking or going from quiet waking to active waking. All right, so I guess I have still a reasonable amount of time. I have about 40 minutes. So yeah, so I'm going to talk about some of my work on particularly the medial septal input. So it would include cholinergic, but potentially also GABAergic and glutamatergic input for grid cells and time cells. So this shows, I've got this going through the speaker, so you should be able to hear. That spiking activity you hear is the kind of a representation of the spiking activity of a single grid cell being recorded. The grid cells obviously were discovered by Edvard and May-Britt Moser in the dorsal medial entorhinal cortex. And this just shows a video of an animal foraging. Maybe the sound is too annoying. I'm going to go back, but it's showing an animal foraging in an open field environment. So similar to what Zach was showing you, it's getting food actually randomly scattered around bits of crunched up Fruit Loops actually, and that keeps it motivated to forage around in the open field environment. You can see this is the location of the rat each time a single grid cell fires a spike. And as you can see here, with it sped up, you can see the trajectory is relatively evenly distributed, but the spiking occurs at particular set of locations in the environment. And they seem they could be described as falling on the vertices of tightly packed equilateral triangles or in kind of a hexagonal pattern. And that was what was so exciting about this discovery, initially up in the medial entorhinal cortex and the dorsal end of the medial entorhinal cortex. So there's been a lot of work on grid cells, but what we've done related to grid cells was to look at the role of the medial septal input in modulating the grid cell responses. And this relates to my long-term love of the cholinergic input, but with that actually motivated me to start working on theta rhythm because of the implications of acetylcholine for theta rhythm and the fact that the medial septum is so important for theta rhythm. So as I mentioned before, acetylcholine and GABAergic neurons are projecting into hippocampus and entorhinal cortex. And what we did in this study was to do infusions of the GABA-A agonist mucimol into the medial septum. And that essentially shuts off these populations of neurons. And that has the effect of, it shuts off the theta rhythm oscillations. You lose the rhythmic inhibition of the GABAergic neurons, and you lose the tonic depolarization of these neurons. And that will greatly reduce the theta rhythm oscillations. So this shows that effect. Here we have theta rhythm oscillations recorded in the field potential of the medial entorhinal cortex in a baseline condition, and then after we inactivate the medial septum with mucimol, you can see robust decrease in the theta rhythmicity in the field potential that then recovers over later times. It's a relatively slow washout for mucimol. And then simultaneously, we were recording a grid cell in a 1 meter square environment. This grid cell has a relatively large space between the firing fields, but you can see this regular spatial pattern. During the medial septum inactivation, you can see we lost this pattern of spatial selectivity of the grid cell. It seems to be firing relatively uniformly throughout the environment. And then the spatial periodicity recovers as the theta rhythm recovers. So it suggests that for the mechanism of grid cell firing, you have to have the medial septal input. All right? It might not be the cholinergic components that's essential, but some component is essential. And I don't have the final answer for you. We've been spending the years since then trying to show whether it's selectively cholinergic inputs, but we tried DREADS first, didn't get that to work, so we've been doing it with optogenetics now, but I still don't have an answer for you on that. So it's an interesting question, but this is a very robust effect, and it was replicated right away by Stefan Leutgeb's lab doing lidocaine infusions into the medial septum. So this suggests a role for theta rhythm oscillations in generating the spiking activity. And this is just showing, I won't go a lot into this model, but just since Trevor wanted me to talk about modeling, we had a model that Neil Burgess had developed. It's a very simple firing rate model with oscillatory dynamics where the frequency of the oscillations is being shifted relative to the velocity of the rat projected in different directions. All right, so the shift of these different frequencies results in the change in phase relative to each other, and this velocity modulation of these different frequencies, then you sum the different frequencies, and when they're in phase with each other, they generate spiking activity that can very effectively replicate the spiking properties of the grid cells in the open field environment. So it's a very nice abstract model of the firing of the grid cells. And it obviously depends upon theta rhythm oscillations, so it could indicate how the medial septal removal of the oscillations would impair the grid cell firing. And we've done, I won't go into all the details of this, we showed that the frequency of the oscillations in the dorsal versus the ventral medial entorhinal cortex is different. And this was testing a hypothesis from the model, a prediction from the model. The prediction was that the cells with a narrow spacing between the firing fields would have a higher intrinsic frequency, and the cells with the larger spacing between the firing fields would have a lower intrinsic frequency. Now I want to emphasize, this is the data from dorsal versus ventral medial entorhinal cortex. So this is done in vivo. These are separate studies done in slice preparations, but overall there's a correlation between the fact that the intrinsic rhythmicity of the neurons is higher frequency in more dorsal regions, which the model could then use to generate narrow spacing between the firing fields. And the lower frequencies are present in more ventral regions, which could be used to generate wider spacing between the firing fields. And this, we've proposed that this actually could also underlie an interesting effect, which is that Caswell-Berry showed in a novel environment, the spacing between the firing fields is wider. And then as that novel environment becomes familiar over multiple recording sessions, they see the spacing actually decrease systematically as the stimuli become familiar. And we propose that this could be related to cholinergic modulation, that the novel environment is resulting in higher levels of acetylcholine release, and that's changing the intrinsic frequency of the neurons, which would then influence the spacing of the neurons. This guided us to do experiments in slices, again, looking at the intrinsic resonance frequency of neurons, which they have a resonance peak. This is giving a oscillatory input that varies across different frequencies, and you see a resonance around theta rhythm oscillation frequency. And then when you put on cholinergic modulation, it actually reduces that resonance frequency. So the idea is that carbachol is reducing the resonance frequency, and if you put that into the model, you can actually generate wider spacing between the firing fields with this, it's the hyperpolarization activated cation current that's mediating that lower resonance frequency, and actually would be slower rebound spiking that we can then use in the model to generate wider spacing, as opposed to the case where the cholinergic modulation isn't decreasing the H current and you would have narrower spacing between the firing fields. So this is another effort to link the cellular level effects of acetylcholine to these network level effects that have been observed in grid cells. So this is pretty much what I already said to you. Are there any questions about this? I know I'm kind of going through a lot of stuff very fast.
You mentioned this, but so what exactly is the purpose of these grid cells? What do they do? Why are they firing?
Yeah.
And if you mess them up, what does it do?
Yeah, so that's a really good question, because they've been proposed to be, many times you'll see it described as being the GPS of the brain. I think that's how they publicized the Nobel Prize for the Mosers. And it's definitely not the GPS for the brain, because for one thing, the rats don't have a link to a satellite system across the world. But other than that, it obviously is a big problem if the spacing of these firing fields changes with novelty. And it's an interesting question. So a lot of papers have shown how you need the different spacings of the grid cells in order to get a spatial location signal out of the grid cells. And it's quite effective if you have all the cells with constant spacing. You put together cells with different spatial phases and different spacings between the firing fields, you can get a really accurate location signal. So that was the initial proposal, was that the grid cells were providing that location signal that then potentially could drive place cells. But this is a problem, because if you don't have a stable spacing of the cells, it's going to mess up that spatial signal. What they proposed, what Neil Burgess and Caswell Berry proposed, was that this maybe is a way for the system to reset the place cells, so that when you're in a novel environment, nothing matches up anymore, you can't get an accurate spatial location, and that would then cause the place cells to remap for a novel environment, and then you would somehow put together the system again. But there's another important thing about this, which is that it's been shown that place cells don't seem to be dependent on grid cell firing. So Mark Brandon, who did the work with me showing loss of grid cells, he looked to see whether place cells would last during medial septum inactivation, and the place cells are still firing, even though the grid cells go away. So it seems oddly, we would never have expected this early on, it looks as if the place cells are driving the grid cells rather than vice versa, which, the scientific data will always kind of throw you for a loop.
When you're going from someplace that's familiar, that has this high spatial frequency, very narrow grids, and then to a novel environment where they're more broadly spaced. When you're going to a novel environment, you have a lot more information to process. And in order to process that information, you think you would have to take things more as a wider swath to get familiar.
Yeah.
Now, when you go, at least in the main part of the hippocampus from dorsal to ventral, when you get to the ventral areas, you get all of these limbic inputs that starts to put an affective bias along the place. And I wonder if that's part of the expansion, is that now you have to integrate all this affective information into place information.
Yeah, no, that's a really good point. And I actually did a commentary on a paper from the Moser lab in 2008 where they showed that the place cells also change their scale, dorsal to ventral, so they get larger and larger really huge place fields in ventral. And I even said, oh, maybe it's a way, and it had been shown previously the ventral was important for fear conditioning, and it might be that there you have a place cell that's essentially coding an entire context-dependent fear conditioning apparatus or something like that, so you can get this. So you can think that maybe there is a way of coding kind of the, affective salience of a particular environment based on this larger scale representation.
This is a fairly obvious point, I'll throw it in here. What about other modulators? What would they do to?
Yes, and in fact, I asked that exact question, and we have a paper in review now at Neuroscience that Caitlin Monahan did where we did 8-OH-DPAT infusions into medial septum and benzodiazepine. Actually, we did both systemic and local infusions. And sadly, to my great disappointment, we did not see any change in the grid cell firing properties. It was really disappointing for me because I was hoping, the biggest reason was I thought benzodiazepines have an amnesic effect very similar to scopolamine. In fact, they can't be discriminated in human studies. So I was hoping that the benzodiazepines would also shut off the grid cells, and they don't. So we don't have a unifying explanation for this amnesia based on grid cells.
This expansion with longevity, is it happening at all the levels of grid cells that are very tight in general and for the ones that are larger? And do they, so the relationship between them, is that kept?
Yeah, that's a good question. They didn't really do, these are really hard experiments and very hard to do multiple grid cell recordings, so they don't have enough data to test that. But that would be one way. If they all expanded exactly the same way, they'd at least maintain consistency with each other. But when they proposed that it was causing remapping, they actually assumed that it was disjunctive expansion. You're asking great questions. I like it because it clearly indicates you're understanding what I'm saying, which is very good. All right, so just to try to get at this question, I mentioned I don't have a final answer for you, but as a first step, we wanted to see was the cholinergic input necessary for the grid cells. And so Aaron Newman, who's now a faculty member at Indiana University, did systemic injections of scopolamine and looked at grid cells before and during and after. And he did see an effect on the grid cell firing. So here's a grid cell recorded before systemic scopolamine, during systemic scopolamine, and afterwards. And there does seem to be a loss of the spatial selectivity of the firing during the systemic scopolamine. But as you know, it's a systemic injection. It could be affecting basal ganglia. You know, it could be any number of different structures being affected here. It could be neocortical sensory input. So we don't have a final story, but it is suggestive that potentially the cholinergic component is important for the grid cells. And we are working, as I mentioned, we're working on testing this now. Holger Dannenberg, who came to my lab from Bonn, he had done work on cholinergic modulation of theta rhythm with optogenetic techniques, and he has been doing studies with archaerodopsin expressed in the medial septum, then shutting off the medial septum, so at least reducing spiking activity in the medial septum. Again, as Martin said, it could be that it's being regulated locally at the terminal, so it might not be the final influence on it, but this is what we're testing. This just shows that you can then record an entorhinal cortex and do the optogenetic inactivation of medial septum. And he has seen with optogenetic inactivation of all the cell classes in medial septum, he can get robust reductions in the amplitude of theta rhythm oscillations. But this was actually done across all the cells, so this is actually, could be due to the GABAergic modulation as well. And like I said, we don't have the final story. We have some effects on the, so we've done, he's done expression. Sorry, the first one I showed you was actually all the cells. This is just the cholinergic chat-Cree cells expressing the archaerodopsin. But we don't have a final word for you. I anticipate that question, so I want to know we're working on it. And he has seen some changes. We see changes in firing rates of neurons with running speed. So it's somewhat related to Zach talking about effects on running speed with the serotonergic manipulations. But this is not so much of a behavioral effect. This is an effect on the coding of running speed by neurons. So there's neurons that will respond to the running speed of the animal. This is a case of a neuron that's systematically decreasing its running speed, decreasing its firing rate with higher running speed. And it's also a head direction cell, so it's responding based on the direction of the animal in the environment, which would be, having both speed and direction coded is an important way of giving that input to the model for generating grid cell responses. And what we showed was this inhibition. In this case, this particular cell seems to lose its speed coding, even though it maintains its head direction coding. And this is actually consistent with the mucimol infusion data, where we showed that even when the grid cell firing was lost, you still had head direction selectivity in the medial entorhinal cortex. I guess I have enough time to, so this is still relating to episodic memory. I've shown that inactivation of the medial septum reduces the activity of grid cells, so that could be impairing memory by taking away accurate spatial representations. And I didn't show you the slide, but mucimol infusions also impair performance in various spatial memory tasks. So the Morris-Water maze, delayed spatial alternation, those are impaired by inactivation of the medial septum. So it could be the impairments in those types of tasks are due to the loss of the grid cell spatial representation. But then another component of episodic memory is the ability to remember the time of events. So, hopefully you can differentiate things that happened at different times in the same room, have very distinct episodic memories for that. You presumably would need some way of differentiating different memories at the same location, even though if you're following Trevor's guideline, you should be moving your chair for each different talk, so you can use spatial code too. But so this is relevant for cells that we refer to as time cells. So here is an example of a hippocampal time cell. I'm going to turn up the volume again. So the sounds you hear are 3 different neurons being recorded in the hippocampus as a rat runs on a treadmill. And it's in the same location, but it's running for a period of about 16 seconds. And for each of these, you hear different frequencies, and you're also seeing different colors plotted. So there's one cell that fired at the beginning of the trial, and this is done over multiple trials, so the cells are consistently firing. This one's firing at one time, this one's firing in the middle, this one's firing towards the end. And you'll see actually the individual cells are also place cells. They're firing at a particular location in the environment. So that's the cell coded in blue that has a place cell firing field here, but now when he runs on the treadmill, you'll see first the cell coded in red, then the cell coded in green, and then the cell coded in blue. So it had a place cell firing field here, but it also has a time firing field towards the end of the running for each of the trials. So I should mention this is work done in collaboration with Howard Eichenbaum. And you can see the green cell also has a place cell firing field. So essentially place cells are coding both spatial location and the time of events, at least in this particular task where we can have repeated trials and statistically test the reliability of the firing.
What have you lengthened or shortened the trials? Is that maintained but sort of stretched out?
Yeah. So I actually took out the slide where I was showing that. We specifically did this on a treadmill. So I want to mention that Eva Pastelkova initially described these types of responses in a running wheel, but she couldn't really change the duration or the speed of the running wheel. She could change the duration, but she said the animals would get pissed off if she changed the parameters, which is very true with the rats. But we did it on the treadmill so that we could change the speed of the treadmill systematically and thereby have the animals running for either different distances or different times. And unfortunately, I took the slide out because I didn't think I had time, but depending on which is the most relevant dimension, you get coding of either time or distance. So if the animal can predict when it's going to end based on time, like it's always 16 seconds, you can have different speeds, so he's running different distances, but it'll always fire at the correct time. If you have the animals running the same distance for different periods of time, then he can predict it based on distance instead of the time interval. So they seem to be able to flexibly alter their firing.
So this is a forced running task?
Essentially it's forced, but we trained them earlier, but yeah, the treadmill turns on and he has to run once it turns on.
Have you tried it with voluntary running?
We have not. I mean, in a sense, the Pastelkova was voluntary, but again, they were highly trained. Yeah, I guess, yeah, not this particular experiment. The speed modulation has been done in foraging in the open field. So I have lots of data on these speed responsive cells, and that's, of course, voluntary.
Does the color date change when the animals are turning like in a clockwise or counterclockwise direction?
Yeah, so Eva Pastelkova argued that the animal had to be performing spatial alternation, and in her case, they did seem to be selective for the direction of turning, but in our study, we didn't. They were primarily firing based on the time. There must have been something different about the design of the task, and we could actually do a version where they would just run to one side and you would still see them firing. Yeah, so this is a very interesting way of coding time events.
So have we thought about whether or not the red cell was firing with the treadmill was starting? They could be also stress response?
Yeah, I mean, well, these are reasonably overtrained animals, so they're not particularly stressed by it. Certainly, the initial thing, there's quite a number of neurons that fire right towards the beginning, and that could be initially coding a stress response like that was generated during the initial training. So yeah, that's certainly possible. And the ones at the end could, you could argue they were initial during training. He wasn't getting reward here, but during training it was getting reward. So that could be like a residual reward response. So I think really these ones in the middle are kind of the strongest evidence for some kind of time selective firing. Though you'll notice there's actually a tendency towards a systematic change from sharper firing at the beginning towards less sharp temporal firing in the middle.
So, in the case where the rat is running on the treadmill, it's getting a proprioceptive feedback that it has traveled a distance, but visual feedback is telling me that it has not traveled a distance.
Yes.
In a case where, let's say, you put it in a virtual arena, where it's physically not traveling a distance, but visually it's getting input that it's traveling, so what will happen then?
Yeah, so we didn't do that experiment, but that has been done by David Tank's lab as well. Well, I guess David Tank's lab did it for grid cells. John O'Keefe's lab has done it for play cells. There's a little bit of controversy in the field, but in general, there have been paradigms where people, at least, I guess on linear tracks, there's no controversy. They can reliably get location-dependent firing that is entirely cued by the visual cues in a virtual world, all right? It's not as robust. The most robust firing is when the distance run matches the visual stimuli. But when you have a mismatch, if the animal's stationary but the stimuli come by, there's still some firing that tends to stretch out. And if you mismatch them, as often happens with these recordings, you get a little bit of each coded.
If they code for place as well as for time, is there any relationship between the successions in space and the time coding?
Yeah, we don't see a system, and you can kind of see that already. We don't see a systematic relationship, and you can see that already in where the cells fired. There didn't seem to be any particular relationship between the place cell fields coded and the time cell. A nice thing would be if red, green, and blue fired in temporal order, and then red, green, and blue fired in spatial order, but that did not seem to happen. The brain always makes things complicated. So these are great questions. I just wanted to lead into the fact that we then looked for grid cells to see if they showed the same sort of responses. Here we're showing cells that are having there's place cells, and some of them are place cells and they have single firing fields. Modeling work that we did suggested that grid cells should have multiple firing fields on the running task. And here's an example of a cell that does that. So this is a cell characterized as a grid cell, and it's showing on each trial it fires at multiple different times. So you can see now here it's firing right towards the end of the trial, and then another trial will start, and we're just jumping to the next trial. So it fires right at the beginning. There's just a couple spikes. Fires in the middle, and then stops entirely. And then it fires at the end, all right? So this is a different cell, but you can see it's actually, and here it'll do it again, fires at the beginning. It's actually before it starts running. Then it's quiet, then it fires in the middle, stops, and then fires again. So this is a cell like this. It's a different cell, but this is a cell that in the open field was characterized as a grid cell with these regular firing fields. And then on each of the running trials, it fires near the beginning, stops, fires in the middle, stops, fires at the end, all right? Now, I'm not going to say the place cells didn't do this. Sometimes the place cells had more than one time cell firing field, but the grid cells with multiple spatial firing fields tended to have more temporal firing fields as well.
Is the time on the treadmill, is it consistent from single to single?
In this one, we were keeping it constant. So the previous experiment, we manipulated it, but here.
Okay, so how do they know if it's like end period of the...
Well, so the animal really knows very well that it's a 16-second time period for running. All right, it's gotten to know it very well in this case.
What if that cell that's firing at these different times, the last one you pointed out, is actually the firing distance, or when it's traveling the same distance without the treadmill, it will fire at these same locations. So do you get what I mean?
Yeah, I see what you're meaning. Is it a grid cell? Does it
same distance on the wheel?
Yeah. So the idea, our original hypothesis was, and I took out that slide as well. We had a model that was Neil Burgess's model of grid cells, and if you just assume that the animal is getting, a distance and a speed signal over time, then the cells that are coded as grid cells in the open field will fire as time cells on a treadmill if you assume they're being driven by the proprioceptive feedback and the direction. And essentially it doesn't match up in the sense that, these guys have relatively regular firing fields spatially, but you can see it's actually starting out sharp and it's actually getting broader. And this was pretty systematic that the firing would get broader and broader. So it's not the case that the animal is essentially coding it as if he were running through a spatial environment, because then you would expect the firing. Well, it wouldn't necessarily, yeah.
It is looking in different directions.
Yeah, that's it. If he thinks he's running, these aren't the best ones, but these are actually autocorrelograms. But imagine it's a grid cell firing field. If you imagine he thinks he's running like this straight through these 3 cells, they should fire at regular intervals, but if he's at a different angle, then yeah, they should fire at somewhat weird intervals. David Tank's lab tried to analyze that for running on running in a virtual world, and they actually argued that there was evidence for that. But our systematic widening of the firing fields, I think it argues against that. But we really haven't managed to answer that question. So it's a good question whether they really are essentially coding time and space somehow in the same space-time dimension and then imagining running through a trajectory.
So say that you increase the length of the trial by 50%, so now it's 16 seconds, so would you still expect to see in this paradigm the 16-second neuron firing also after 6 seconds?
Yeah. It's a really good question, and we didn't do that manipulation. We somewhat answered it with the manipulation, the previous one in hippocampus, where the cells, if it was the same distance of running, they would actually fire at different times. And so that suggests that they weren't locked into a particular time of running. But the problem is that the animals were either in a distance manipulation or a time manipulation, and we really should do like a surprise change when they're mostly doing something else. And we haven't really done that to systematically test that.
I'm so confused about the function of the grid cell. So what do you think it signals? Is it like only if you're on a patch that you know where you are, either in space or in time, or is it more like a confirmation that, oh yeah, at least I'm here?
Yeah. I think they are important for coding of space and time, and that's consistent with tasks showing entorhinal lesions will impair performance of spatial memory, and medial septum inactivation will impair spatial memory. There hasn't been as much work on this, but I think Pousset's lab had actually looked at the ability of animals to judge time intervals, and I believe they did it with medial septum inactivation. I've only seen it as a talk. And I think they have evidence for impairments of time interval coding with medial septum inactivation at least. Now that's a little bit tricky because that's influencing hippocampus as well. So the models, especially the initial models, assumed that it was coding space, and also in papers I wrote in 2008, time. So that's like the first existing hypothesis. But then there are complexities such as the change with novelty and the widening of the firing fields that are problems for that hypothesis. We don't have the final answer.
So just maybe along that question, if there was a sealed event that occurred at one particular point in the trial, are these the cells that help the animal know at which point in the trial it's occurring?
Yeah, so that's one idea that I did in certain types of models. The idea, and I was talking about that actually earlier, somebody asked questions during the break. I think that probably, if it is episodic memory, then an important component is to, and I talk about this in my book in 2012, the idea is that you would have some representation of a spatial temporal trajectory, and then you're associating events with positions on that trajectory. So if I walk out of the room and talk to somebody right out in the, this antechamber, I would have like a code of, oh, I remember that was the event that happened in that location at that time. So that you can model that effectively in this framework, but we haven't fully proven it.
So I'm not sure I'm understanding the heat maps correctly, but is it the case that the more dense sort of grid cells, as in when they're on the treadmill, are there more peaks throughout?
Yes.
Is there a relationship between the two?
Yeah, so that was a prediction of the model. Unfortunately, we didn't have enough data. In this experiment, we tended to really only get 2 different spacings. You can sort of see them, slightly narrower spacings here and then slightly wider spacings. So we really didn't have enough data to prove that statistically. So that was a hypothesis, but it's as yet unproven.
question, like, is that possible that grid cells could, what can I say, evaluate or just encode also speed cells?
Yes, and in fact, I used this in my model. I actually said that in the model I published, you could code not only your episodic memory for events at particular locations and times, but your speed of movement at particular locations. And I think that's an interesting hypothesis, because that's, Tulving doesn't talk about it in his initial definition of episodic memory. So I saw that as something different, and intuitively I think that makes sense, but I don't know of any data yet on that for episodic memory.
Another question is, when do these cells start firing? So like, do they have to learn the task well, or from the very get-go they already have this pattern?
Yeah. So for this one, with running on the treadmill, we had them learn the task before we recorded them, because it's a difficult task. But the Mosers, well, in any case, the answer is that if you get a new cell, it's going to fire in this case, but they're overtrained. But the Mosers actually have done manipulations where they look at the firing of a grid cell the first time in animals in an environment. And they seem to see that if they correlate where it fired on its first trajectory, the firing fields seem to match where the long-term firing fields are established. So they think of it as it can change its locations in different environments, but they think of it as a map that's kind of already right away created. These are great questions. So just the bottom line of this time cell data, and what I find the most exciting, is that medial septum inactivation impairs grid cell spatial selectivity. It also impairs time cell temporal selectivity. And this is something that Eva Postelkova's lab has published in 2014, right? So she initially described the time cells in a running wheel. She doesn't call them time cells, she calls them episode cells, which is a perfectly reasonable, I didn't come up with the term time cell either. Howard Eichenbaum came up with that term. So I'm kind of neutral about it. But in any case, she looked at what we call time cells in the baseline, and then during medial septum inactivation, this one completely shuts off, and you think maybe it's just a lack of drive, and then it recovers afterwards. But here's a cell that has 2 firing fields. So this is actually a hippocampal cell with 2 firing fields. And then during medial septum inactivation, it's still firing, but it loses its selectivity, and then the selectivity comes back. And this is very exciting to me. So it does suggest that there's a unified mechanism for the firing of the grid cells and the time cells, which is consistent with, well, it's consistent with a couple of different models. So it doesn't yet select between all the different models, but it's exciting that the models, in a sense, predicted this. And you can see here's a whole array of time cells. This is getting to be a common way of plotting the data. These are cells recorded at different times, but they're just ordered by what time they were coding across many trials, and then they lose that selectivity of coding during medial septum inactivation, and then they regain that. So this could also be a cholinergic dependent mechanism. All right, so I'm actually going to get to the last section of my talk, which is great. It's exciting because I really like the video that I get to show. And part of the reason I like this video so much is that it shows how tremendously important acetylcholine is. So I know it was only 5 out of all of us that thought acetylcholine was our favorite, but hopefully this will maybe recruit more people, because it shows if you block dopamine or you block serotonin, you don't wipe out cognition entirely. But if you block muscarinic receptors, you essentially destroy cognition. All right, so this is relating to the different levels of acetylcholine during waking, quiet waking, slow wave sleep. I already showed that to you. Again, it's just assuming kind of a tonic change in these different states. And as I mentioned, the muscarinic presynaptic inhibition is changing relatively slowly, which would be consistent with a slower change in modulatory state. And this fits with an existing body of theory. So Yuri Bajaki initially proposed this 2-stage model of memory formation, and it's also been studied quite a bit by Bruce McNaughton and Matt Wilson and Jay McClellan. So the idea is that Yuri Bajaki initially proposed that during waking you encode information in the hippocampus, and I'm calling it an intermediate term episodic memory because I want to differentiate it from the long-term episodic memory that's still maintained in patient HM, for example. And then the idea of this model is that during slow wave sleep or quiet waking, there's this consolidation process where the consolidation involves a transfer of the representations from the hippocampus to the neocortex to form long-term episodic memories and potentially build up semantic memory. So this is a very well-known neuropsychological theory. And it's consistent with some of the data on patient HM and so on. What I'm going to talk about is the role of acetylcholine in changing these dynamics. And this is something that Yuri Buzaki talks about right away in his 1989 paper, because he shows a change in the dynamics of the circuit with atropine, with blockade of muscarinic receptors. So I'm really just taking his idea and showing how it relates to the circuit level effects that I talked about. So the basic idea is that during waking you have high levels of acetylcholine, and this could enhance the feed-forward, this enhances the feed-forward influence of neocortex on hippocampus, but reduces the influence of the hippocampus on the neocortex. And then during slow-wave sleep or quiet waking, the hippocampus is very robustly active during short periods of time, and the lack of acetylcholine, especially during slow-wave sleep, means that it'll have a very strong driving influence on the neocortex. And this is consistent with this idea that cholinergic modulation is normally suppressing the excitatory feedback. So normally during waking, you would have higher levels of acetylcholine suppressing the excitatory feedback in CA3, from CA3 to CA1, from CA1 back to the neocortex. And I mentioned this early study by Winston and Abzug, who actually showed during waking, if you stimulated the inputs, you'd actually see much less activity at the output, like in the subiculum. And if you did it during slow-wave sleep, which would correspond to this, if you stimulate the input during slow-wave sleep, they actually saw, way back in the 70s, much more robust spread of activity through the circuit, consistent with the fact that there's no longer this presynaptic inhibition, so each stage is much more strongly driving the output structures. And this is also then consistent with what Yuri Buzaki and Bruce McNaughton have studied, which is the spread of activity back from the hippocampus to neocortex. So in a very simple model, the idea is that during waking, you have information coming into the neocortex, but the synaptic modification in neocortex is relatively slow, so initially it's driving hippocampal cells, and you have this more rapid LTP induction in the hippocampus, all right? Following exactly the same framework I already showed you earlier, but it's not going to really strongly drive the neocortex back, and I'll show you why it's important that acetylcholine be regulating these connections. That's in the video.
I kind of asked earlier, but I'll ask in a different way. So do you think that the mechanism of active walking in terms of acetylcholine is the same with memory information? So the same mechanism, but different amount of acetylcholine, or different mechanisms?
Yeah, it's a good question, and we don't have the answer for that yet. Because it's the easiest way to change the state in animals, we've tended to focus on the walking state. And in fact, I have to say I tried for years to come up with a good systematic way of activating acetylcholine in stationary animals. We did fear conditioning. We were measuring theta rhythm during fear conditioning, and we saw effects, but didn't really see systematic cholinergic effects. And we tried novel stimuli as well, and even though you see the microdialysis change, we didn't manage to systematically activate it that way. So it's an open question. But at least, humans just during, and animals, during active waking, it isn't just, the microdialysis does show it isn't just when they're running, it's also attention to stimuli that will cause this higher levels of acetylcholine release. But then the idea is when the acetylcholine is much lower levels during slow-wave sleep, now you've released this feedback, this presynaptic inhibition of the feedback. And so if you give there's no sensory input, but you give any kind of random input to the hippocampus, and you'll actually, once you activate this cell, it'll actually tend to activate the other cells in a particular memory, and then they will together drive the neocortex. And the idea is that over time, this process will gradually modify the connections in neocortex so that the association initially stored in the hippocampus will now be stored in the neocortex. And maybe you've even had enough time to consolidate, the memory of the association of leather and boot or something like that you can already remember it if I had your, well, it wouldn't happen now. You'd have to probably have a night's sleep before it would get coded in the neocortex. But in any case, so this shows, again, that these different levels are present. This difference in the excitatory feedback is consistent with what Yuri Buzaki initially described. Well, actually, these were discovered initially by John O'Keefe, these sharp wave ripple events. So how many people have heard of sharp wave ripple events? It's a subset of people. So these are basically very robust events that occur when an animal is sitting quietly, or often when they're drinking or eating some food reward. Instead of seeing theta rhythm oscillations, you'll see kind of more irregular activity in the EEG, and then suddenly you'll see these big sharp waves in the field potential in stratum radiatum, and they're coupled with these high frequency, what are called ripples. They're quite high frequency, like, you know, on the order of 150 to 300 hertz. And a lot of studies from Bruce McNaughton, Matt Wilson, Lauren Frank, Shantanu Jadhav have shown that if you look at the place cell firing fields during the theta running on a track, so this is a running on a track with relatively long intervals of seconds over this running period, you can look at a sequence of place cells, and then you look during these sharp wave ripple events. Now this is a very different time frame. This is, I guess I didn't put that in there, but in any case, these are milliseconds apart from each other. So these are all within a 100 millisecond period, and they're firing in sequential order. So it's this idea that you're encoding a sequence over many seconds, and then you're reading out a compressed sequence during these sharp wave ripple events that's much shorter. This is, you know, maybe takes 5-6 seconds here, takes maybe 100 milliseconds to read it out during a sharp wave ripple. And that's exactly what you would need if you needed to consolidate a previously encoded sequence. So that's what might be happening there. You encode the sequence with the slower waking dynamics, and then during these sharp wave ripple events, the sharp waves could be essentially driven by the very strong recurrent connections in, you know, the sharp wave can be driven by these recurrent connections and the connections from CA3 to CA1 that have this sequence encoding in them. So that's the basic idea. So it's really just showing how the acetylcholine effects are consistent with this. And as I mentioned, Yuri Bajaki had already shown that giving atropine will increase number of sharp waves. So you're blocking that presynaptic inhibition. They've shown also recently that if you do optogenetic activation of cholinergic neurons, it reduces the number of sharp waves. So it's really good data supporting that. And there's really interesting tests that Jan Born did in humans where they tested these hypotheses that I described in the 99 paper. One hypothesis is that if you increase levels of acetylcholine during sleep, it should actually impair consolidation. And they showed that. They gave physostigmine before people went into slow wave sleep. They showed impairment of consolidation. The other is that if you block acetylcholine receptors during waking, it should enhance consolidation. And they showed that as well, that if they had subjects learn stimuli and then instead of just sitting quietly, they actually got scopolamine, they then performed better. And in fact, that's consistent with what I told you about the Goneim and Maywalt study, where they actually had slightly better retrieval after the scopolamine injections. All right, finally I'm getting to my video, and I've got plenty of time to show it. So I said I would describe to you why is acetylcholine so important? You know, both I want to kind of say it in connection to this model, but I also want to say, well, you know, just as a sort of a advertisement for acetylcholine, like I said, if you have really high doses of blockade of muscarinic acetylcholine receptors, you not only are impaired in your memory, you really have a total breakdown of cognitive function. And I'm going to show you this video of this. This is not a controlled experiment. This is an experiment run by people on themselves. And I will skip the intro, but it's due to something called Jimson weed. And this is a guy that, for interesting reasons, decided to make a brew out of Jimson weed and then drink it.
Very close attention to what you're about to see is to treat Cuba Vista policeman and James and his friend under the influence of Jimson weed. The two are so impaired they can't even speak. Again, what you're about to see is no act. James is the one on the right. James and his friend are truly in a world known only to them. Here you see them pass a cigarette to each other, only there is no cigarette. James' friend later smokes it and actually flicks the ashes. Again, there is nothing in his hand. James styles his hair with an imaginary comb. He beckons to friends in the room that only he can see. There is fear in the eyes of both boys, and major anxiety. Motor coordination skills are almost completely gone. seen it before.
Okay, good.
So I love that as the final point. So he doesn't remember anything about it at all. So essentially, this is what happens if you block this cholinergic, and it's not obviously not just that, but I like it because essentially it's a nice piece of anecdotal data supporting the idea that if you block the muscarinic receptors, you essentially have consolidation mechanisms gone wild, all right? They're causing hallucinations. I think that what it is that there's previously stored representations in their brain of a cigarette smoking, seeing a friend in a corner that are getting activated, and they can't differentiate because this influence is so strong, it's activating the neocortex and creating this imaginary hallucinatory world at the same time as preventing them from encoding new information. So, so that's where I'm going to conclude. Thanks.